A-Level Biology 9700, Paper 4, Variant 3, May June 2024, question number 3 and question number 4. And question number 3 is, in plants and humans, the phenotype of an organism is determined by the genotype and the environment. Plants from the genus Primula have different petal colors. The presence of the pigment molybdenum results in blue petals. So the metabolic pathway for molybdenum synthesis is controlled by gene capital T small t, and the presence of the dominant capital T results in blue petals. So it means the molybdenum is a pigment which gives the blue colors to the petals. So due to the gene uh, capital T small t, and capital T is responsible for this formation of pigment. Capital T is a dominant allele. Another gene, capital D small d at a different locus, also influences the molybdenum synthesis pathway. When the dominant allele D is present, capital D is present, its gene product suppresses the molybdenum synthesis pathway. This is summarized in figure 3.1. And figure 3.1 is precursor allele capital T formation of molybdenum, which gives the blue color to the patterns. And precursor allele capital D, no molybdenum production, and due to which non-blue patterns. It means capital T is responsible for the formation of molybdenum and the color blue petals. On the other hand, the capital D is also a dominant allele, which will prevent the formation of uh, molybdenum, and as a result of which non-blue petals will be formed. So in this way, we have come to know that what is happening over here. It means the capital T is influenced by the capital T, due to which whether it is in capital form, but it will be unable uh, in the formation of molybdenum, and the result will be the blue petals. So in this way, the capital D is influencing the capital T. So question is, a genetic cross was carried out between two plants, heterozygous at both gene loci. The resulting offspring's genotype are shown in the Puneet scale in figure 3.2. And in front of you, there is figure 3.2. And now according to this, we have to find the answer. So we are moving towards the question. After this figure 3.2, state which of the genotypes shown in figure 3.2 have blue petals. So keep in mind, already we have discussed in question that capital T is responsible for the formation of uh, molybdenum, due to which the colors will be petals will be of blue color. Whereas the capital D, it will stop the formation of molybdenum, due to which the colors of the petals will be uh, without a blue color. So in this way, we have to find out the petals which are of blue colors. So the genotype for them is, so both are capital, so it will not be for no, no. Wherever the capital D is there, there will be no formation of blue petals. Capital D, capital D, capital D, because it is a dominant allele. So dominant alleles will not be able in the formation of colored patterns. So it means, and here there is small t. So again, they will not produce the blue petals. So here you can see the two genotypes, capital T, small d, small d. So small d will not be able to prevent the formation of molybdenum, due to which the color of the patterns will be blue. And same is the case in the other type of genotype. So 1, 2, these are the two genotypes which are responsible. These two are the same genotype. So two genotypes are there which are responsible for the blue petals. So I would like to write them capital T, small d, and other one is capital T, small d, small d. So T, whether it is in heterozygous condition, will be able to produce the molybdenum, and small d, they are not in being a recessive allele, they are not able to produce the molybdenum. State the ratio of the non-blue to blue petals. For the so in this cross, you can see that there are the number of genotypes uh, which are producing the non-blue petals is greater as comparison to the blue petals. So we are going to count their number 1, 2, 3, 4, 5, and 6, 7, 8, 9, 10, 11, 12, and 13. So the non-blue petals are 13 and the other petals 1, 2, and 3. They are 3. So the ratio between non-blue petal to blue petals is 13 is to 3. So there will be 13 non-blue petals and there will be 3 blue petals. So name the type of gene interaction that has caused the offspring ratio you have stated in the previous question. Okay, students, here you can see that the both are the dominant alleles, the capital D and small t, but one of them is influencing the other. So this phenomena is a, you can say this is a genetic phenomena, and where the expression of one gene is influenced by the expression of one or more other genes. So in simpler terms, we can say that it's like a genetic interaction where one gene can mask or modify the effect of other gene. So the phenomena according to which it is happening, it is called as epistasis. So students, the epistasis is that genetic phenomena in which the one gene is affected, the expression of one gene is affected by the influence of the other gene. So we are moving towards the next question. Gene capital T, small t, and capital D, small d codes for protein that are involved in the control of the production of molybdenum. Discuss the possible roles of the protein coded for by genes capital T, small t, and genes capital D, small t in the control of the production of molybdenum. So molybdenum, you know, this is a pigment and which is responsible for the red, purple, and blue colors in many plants, including fruits, vegetables, and flowers. So the production of molybdenum is a complex process involving multiple genes and their encoded proteins. So the genes capital T, small t, and capital D, small d are likely involved in regulating different steps of the molybdenum biosynthesis pathway. So now we have to discuss about this. That is, after this explanation, now I would like to write the points which I have discussed with you. So first of all, I will start with this. That is the capital T and D are the dominant alleles which results in a functional protein. The capital P or small t protein coded by capital T or small t would be a transcription factor. It might activates enzyme that catalyzes specific reaction for the synthesis of molybdenum. Whereas capital D would codes for an inhibitor enzyme which inhibits the reaction by binding to enzyme and due to which the reaction will not take place or stop, and molybdenum will not produce. So now we are moving towards the next question. And next question is, now for this question, we are going to explain the relationship between the factor 8 gene or F8 gene and factor 8 and condition hemophilia. And students, you know that the factor 8 gene plays a crucial role in the blood clotting. It provides the instructions for producing factor 8, a protein which is essential for the blood clotting process. So when this gene is defective or missing, it leads to a deficiency of factor 8, resulting in a condition called hemophilia. So hemophilia is a genetic disease. In this condition, blood cannot properly clot. So the excessive bleeding, you can say, due to even from minor injuries, the person can bleed to death. So, you know, the severity of the hemophilia depends on the level of factor 8 deficiency. Individuals with severe hemophilia have very low levels of factor 8, leading to frequent and severe bleeding episodes. And those with moderate hemophilia have slightly high levels of factor 8, and bleeding episodes are less frequent and less severe. Mild hemophilia is characterized by even higher levels of factor VIII, and bleeding episodes are usually only triggered by major injuries. So, in this way, uh, all they are related with one another. So, now I'm going to write this explanation. And students, the F8 gene is located on X chromosome. So it is called as sex-linked genes. It contains the code for synthesizing a protein called coagulation factor VIII. The gene has two alleles, one dominant, which codes for normal factor, whereas the other is recessive allele, which results in lack of factor. So the persons will not having this problem do not clot their blood, and they have hemophilia. Abnormal forms of F8 factor VIII will be less less factor VIII or no factor VIII. So the severity of this hemophilia will be different in all these cases. When there will be no factor VIII, it means the person will have the different scenario as comparison to other when there will be production of less factor VIII. So in this way, the person, if he or she is suffering with this, can bleed to death even sometimes. So in case of severe injury, so this factor VIII is located on the X chromosomes, and in the females have two X chromosomes. So in that case, if she has, so she will be normal. And the other condition, when she will be heterozygous for this capital F, and small f will be, so she will also be the normal, because capital F is dominant. And on the other hand, when she has both small f, then she will have hemophilia. On the other hand, in male, they have only single X chromosome and one Y chromosome. So in case they have capital F on X chromosome, they will be normal. And on the other hand, if they don't have, then they will be with hemophilia. So they don't have any heterozygous condition in this case. They will be hemophilia. Now we are moving towards the next question. And next question is question number 4. And question number 4 is, genetic engineering is a technique used to modify the genetic material of a specific organism to change a characteristic. So genetic engineering uses specific enzymes and commonly involves the use of plasmids for the transfer of genes into an organism. And students, what are plasmids? And then four enzymes that are used in genetic engineering techniques involving plasmids are transcription endonuclease, DNA ligase, and DNA polymerase, and reverse transcriptase. So first of all, I uh, would like to discuss about the genetic engineering. And genetic engineering, is a technology that allows scientists to alter the DNA of an organism. This can involve changing a single base pair, deleting a region of DNA, or adding a new segment of DNA. So in this way, genetic engineering play a very, you can say, great role in this field. And here you can see the four enzymes are given. And what are plasmid? Plasmids are the circular DNA which are present in the bacteria. And in the bacteria, it is prokaryote because it don't have proper nucleus. So DNA is embedded directly into the cytoplasm. And beside this is a specific type of circular DNA is there, which is called as plasmid. So in this case, in genetic engineering, the bacteria play a vital role. They act as a vector because they have plasmids, and they play a role in this genetic engineering. So during this process of transfer of genes into an organism, then the four enzymes are involved. Four different types of enzymes are involved, and outline the role of these enzymes. Now we are moving towards the writing the role of these enzymes. And here you can see outline the restriction endonuclease. The plasmid, when plasmid is used for this purpose, so there is need to cut it down. So to cut it down, so at this case, uh, enzymatic scissors are used for this purpose, and the enzymatic scissors are called as the restriction nuclease. So it will act as a enzymatic scissor. So it will cut down DNA at specific sequence known as restriction site. So this enzyme will be used to cut the DNA or the plasmid. At a specific site. Specific site means the genes which are required for that purpose. Specific site, we will say specific site, or you can say the restriction site as well. So it will act as a enzymatic scissor. And second enzyme, after cutting it down and inserting the uh, say now this it will become in this way, and then after cutting it down, and then what will happen, we have to insert the required DNA into it. So after adding the required DNA, there is need to stick with on both ends. So for this stick, this DNA ligase will act as a glue. So it will act as a glue, and uh, then it is used and used to join DNA fragments together. So, and the next is a DNA polymerase enzyme. DNA polymerase enzyme is this enzyme synthesizes the new DNA strands using the existing DNA as a template. And the other one is the reverse transcriptase enzyme. So this enzyme, uh, which is called as reverse transcriptase enzyme, synthesizes DNA from an RNA template. Students, you know what happens in reverse transcription. In transcription, DNA is copied, act as a template stand, and there is formation of messenger RNA. So in reverse transcription, you know, opposite that is the DNA enzyme DNA from the RNA template. So in this case, the RNA will act as a template and formation of DNA will take place. It will be, you can say, reverse of the transcription. So the enzyme which is used for this purpose is called as reverse transcriptase enzyme. Now we are moving towards the next question, and next question is why promoters as well as the desired genes is often transferred into an organism. And students, you know that when we insert the organisms from one organism to the other, as for example, in this case of the genetic engineering, that one organism is, you can say, inserted into the other, just like the promoter, or you can say um promoter, or any desired gene. So what is the purpose for this? So this can be, first of all, we can say, to initiate the process of transcription, and then you can say it can done because transcription initiation takes place only by the promoter, because a promoter is a specific DNA sequence that acts as a binding site for RNA polymerase, the enzyme responsible for transcription. So, the RNA polymerase recognizes and binds to the promoter, initiating the process of transcribing the DNA sequence into messenger RNA. So, without a promoter, RNA polymerase cannot recognize the gene, and the transcription cannot stop. On the other hand, it also controls the gene expression, so the promoters can regulate the level of gene expression by controlling the rate of transcription. So the different promoters have varying strengths which can affect the amount of protein produced from the gene. So in this way, it has its specific role. Now I would like to write shortly all this explanation. So that is, first of all, I will start from that promoter is like a switch. Is like a switch which turns on the gene. In both ways, it can switch off as well. So the promoter provides a binding site for RNA polymerase enzyme, which is responsible for transcribing DNA into RNA. Promoters can be regulated by various factors. And regulates the gene expression at the right time, by controlling the rate of transcription. So students, first of all, listen my explanation, which I have done before writing this answer. So always listen carefully, because first of all, in every question, I explain the answer, then I write shortly on the given space. Now the next question is, the production of insulin by genetic engineering involves the use of plasmids and the bacterium Escherichia coli. Multiple Copies of genes that codes for insulin polypeptide are mixed with gut plasmids during the process. Only some of the plasmids that are taken up by the host bacteria will lead to the expression of insulin polypeptide race. So figure shows a cut plasmid and the gene coding for the insulin polypeptide, three different plasmids that have been formed as a part of the genetic engineering. Here you can see the promoter gene is there, marker gene is there, and complementary sticky ends ready for gene introduction. And on the key, the gene that codes for the insulin polypeptide. So the direction of transcription is from left to right side. So here this is given. Now they have given us the plasmid X, plasmid Y, and plasmid C. And the question is, comment on whether a bacterium will produce the insulin polypeptide if it has either plasmid X and plasmid Y or plasmid C, and explain the reason for your choice. And first of all, I'm going to this question, then I will write the answer in the given space. So here you can see, uh, compare the X, Y, and Z with the this one. You can say in this way. You can see, first of all, the plasmid X, and you can see in plasmid X, when we insert the arrow is in the opposite. The arrow should be in this side. They have given this, so it should be in this side, but it is in the opposite. It is going in the opposite direction, so it will not produce the insulin because the arrow it should be in this direction, and you can see the arrow is in opposite direction, due to which it was moving in the backward direction, on the wrong way, you can say. The plasmid Y, it has promoter, it has marker gene. So the gene that codes for insulin polypeptide is also going in the, so plasmid Y will produce the insulin. And the now plasmid C, it will not produce the insulin because the, where is the gene? This gene should be inserted into it, but it is not inserted. It means it is also not going to produce the insulin. So only plasmid Y is in a true form, is actual form with by which we can manufacture the insulin by the process of genetic engineering. So now I'm going to write no formation or synthesis of insulin because the gene is inserted by the wrong way, by the wrong way or in the wrong direction, you can see. And why? Insulin production will take place everything is in correct sequence. Again in Z, no insulin formation because required gene is not inserted. So it was all about question number 3 and 4. And please like, share, and subscribe my channel to watch more videos. Thanks for watching.